It's the time. So, hi everyone, welcome to Lab 8. And for this week, we are going to talk about the software as the beta and also the basic data format for conducting research in statistical genetics and the VCF file, and also doing the ChIP-seq motif analysis, which you could encounter all the problems in homework 4. But before our lab for this week, I'm going to make a few announcements. The first thing is about our final exam. So the final exam is around Monday, May 13th at 9 a.m. to 12 p.m. And this is if you are a graduate student, you could also choose the alternative one, which is around May 7th. And if you want to do so, please email Shirley as soon as possible. Sorry that we cannot hold the currently we cannot hold the alternative exam for the undergraduate student due to the school's policy. And if you are an undergraduate student and you still want to do the alternative exam, please email Shirley as soon as possible. And you also have to get approval from our registrar's office. So yeah, if you want to do so, please do it as soon as possible. Then we can go back to our lab material and also more announcements. I wouldn't hold additional office hour for this Friday due to the open house event, and homework 4 is due on this Sunday midnight. So for this lab, basically have three parts to go through. The first part is about the beta. We would so far you have already encountered how to analyze the differentially expressed gene sequencing data, such as using the lemma to conduct the related analysis, and also how to find the transcription factors binding sites, also the motifs, by using the Max2 or some more similar softwares. But how could we combine them all together to conduct some further analysis? One question one might encounter is that which kind of transcription factors might regulate some downstream genes expression, such as being as an activator or a repressor, or didn't have any change at all. So that's basically what beta is doing. We would get from this lab, we would take you guys to have an overview of this software and also its algorithm and its workflow. But since this part is not incorporated into our homework, I would go through it very quickly. And then it's a new area. I think Shirley has already opened a new area about the statistical genetics topics on this Tuesday. So I will talk about it more in next week, but the basic thing is to get familiar with the basic data format, which is the VCF file. And even though it's I would talk about that later. And then for the homework part, I would go through all the questions with you guys to see if you have any problems or any new thoughts. So yeah, as I mentioned 1 minute before, the motivation for beta is quite intuitive and simple. So given the peaks code from the ChIP-seq, we are interested in assessing which genes are regulated by the given transcription factors or the chromatin regulators. Yeah, you can see from this homework, you are focusing on the AR, and we still have a lot of interest in a broad range of other transcription factors and other regulators. So a simple method would be to assign the nearest gene or the gene containing peaks in its promoter region, but ChIP-seq experiments are often examined in the context of gene expression, and thus expression profiles are available for both the factor bound and factor unbound conditions. So how can this information be incorporated with the peaks to improve the inference about the target genes? Actually, it is answered by the beta. So this software is also developed by Dr. Liu's lab in around 2013, but unfortunately, currently it is under maintenance, so we didn't incorporate it into our homework 4, but you can play around from the web page of beta, and I think they have a web server here, and if you can click here, yeah, sorry, it's under maintenance, so just feel free to play around it. And you are highly suggested to check the original paper for the details about the algorithms. So now let's go through all of them. So basically, it is a combined analysis for all the knowledges we have learned so far. The first thing, just think about it. What would you do if you got the differentially expressed data and also the ChIP-seq data? Well, the first thing might use the lemma to code the genes up or down regulated ones, right? And when you got the differentially expressed genes, what's the next step? You might use the Max2 to help you to identify the transcription factors binding site, also the transcription factors binding motifs. That's what have been done by the Max2, which is the first part of our homework. And then combine all this info, or combine these two types of data. We could do some combined analysis by using beta to see whether factors has activating or repressive function, or whether it could identify direct target genes, or to help us to identify binding motif and collaborators. So even though the original designing group has divided the beta into three stages as the activation and repression prediction, the directed target prediction, and the motif analysis, I would summarize them as two parts. So the first part is about combining analysis of the ChIP-seq data and the differentially expressed and the gene expression data, and the second part is to conduct the motif analysis. So here's a basic summary for the beta workflow. The first step is to use the lemma to detect up or down differentially expressed genes and store the result as a CSV file, and then the output genes in three categories: the upregulated one, the downregulated one, or the unchanged gene expression data. And then by incorporating the Max2 software, we could find the binding events or what we call as peaks called from the software and stored as the bed file. And based on these two types of data, we could combine them all together to via beta to see whether this transcription factor has activated or repressed the target gene's expression. Well, I would jump the details about the algorithms. Please feel free to check the original paper. And if you have any questions, please feel free to shoot me an email about that. So the I mean, the take-home message for this lab might be how to interpret the results of the beta. So just think about it without any supporting information. What's your expectation for the analysis of beta? Well, it might tell you the target transcription factors could be potential activators or the repressors, or both activator and repressor, or none as the activator or nor the repressor for target genes expression data. So, for so. Uh, under what kind of scenario could we call the transcription factor as an activator? So from this plot, this is a part of the beta. This is a toy example of the beta's analysis results. So this plot tells us that from this plot, you can see that the red line represents the upregulated genes expression result, and the blue line, or the purple one, could represents the downregulated genes expression result. And you can see that there is a statistics followed by this annotation, which is actually the p-value. So how could we generate this p-value? So basically, we compared the upregulated expression result towards the background, which is the dash line here. And if it's divided far away from the dash line, we call it as the upregulated one. If it's quite near to that one, we see that the upregulated phenomenon is not quite obvious or statistically significant. So from this plot, you can see that the downregulated expression line is very close to the dash line, and the p-value is not quite significant, while the upregulated line is far away from the dash lines and with a very significant p-value. So we might call that this kind of activator, this kind of transcription factor, sorry for that, this kind of transcription factor might be considered as an upregulator or an activator. And the opposite scenario might happen while we switch the upregulated line and the downregulated line. So if we switch them, the downregulated one is significantly far away from the dash line, which is the background here. So compared to the nearest upregulated line, we would call that this kind of factors might be considered as the repressor. And you can imagine that there are two other possibilities where both the downregulated line and the upregulated line might interact with each other. So under the first scenario, they might be interacted with each other and far away from the dash line, which is the background. So under these circumstances, we would call that they might be considered as both the upregulated one and also the downregulated one. So in other words, we would call the transcription factor as both the activator and the repressor for the target genes. But another possibility is that these two lines interact with each other and they are not far away from the background, which means these three lines interacted with each other. That means we didn't have a very significant result from the beta, so this target transcription factor may not be called as the activator nor the repressor at that time. That is not for everyone's expectation, and we don't want that kind of thing to happen. So beta also have a second part, which is a binding motif analysis. It could give you all the details about the motifs that you want to analyze for the target transcription factors. Yes, all these things could be done on the website. And post-beta analysis, we would incorporate the software as the David, also something like the timer, which is sub-project of the Systrom program. We would touch that later, and also do some collaborating factor analysis by using all the softwares listed in question 8 from this homework. So just a brief summary, beta is composed of beta basic, beta plus, and beta minus. These are three parts of this software. So the beta basic can be used to predict whether a factor has activating or repressive function and detected direct target genes, which we have, so the result we have already seen in a few slides before. And the beta plus can be used to predict whether a factor has activating or repressive function, whether it can detect target genes, direct target genes, and whether it can analyze sequence motifs in target regions. And beta minus is used when only binding data are available to predict the target genes based on distance. So both binding and differential expression data are required for beta basic and beta plus. Yeah, that's our original motivation. All right, then let's move to a new area, which is the statistical genetics, and I enjoyed it a lot. But before we go deeper in next week, we have to go through some basic data format for this lab. So the very basic one would be the VCF file. Basically, similar to the BET file, VCF is still a text file format. Remember where we can, if you can remember where we find the annotation and the documentation for the BET file, you can type UCSC data file format in the Google and check this web page. So they get a summary about all the basic data format here. And if you could go to the, yeah, we have already talked about the BET, and I'm going to talk about the MAF next week. And this week we are going to talk about the VCF file. And if you can go to this VCF call format, they got two summary files at here. And if you have enough time to do that, yes, you can see that they have the details about the VCF file and also some toy examples about the VCF format, and also the explanations about all the thresholds or parameters that would be appearing in this VCF file. Okay, let's take a glance at the real example of the VCF file. So the VCF files contains meta information line, with each line containing information about a position in the genome. Let's take an overview of the data file. So this is an example from my lab, and this is actually a result from the whole genome sequencing analysis. So basically, we stored all the whole genome sequencing analysis results into the VCF file, and from the very beginning, you can see the title of the VCF file, which stored the information about the whole genome sequencing situations, and then followed by this, the first 8 columns are basic informations, and they are solid for each VCF file. The first column is the chromosome, and the second column is the position, and the third column is the ID. Well, sometimes you don't have the ID. And the fourth column is the reference allele at that position, and the fifth column is the alternative allele at that position. So here's the tricky part for the VCF file. As you can see that for the VCL file, each row represented each row represented a specific position, a unique position. But the possibility is that at this specific position, it could be encoded with multiple alternative alleles. So, for example, at this loci, you got one reference allele while you have three alternative alleles. So it gives the computational guy a lot of troubles how to encode this allele information into the computational language, and just give you more hints about that. So basically, sorry, I didn't get a marker on this on this computer. So basically, I can only use this. So basically, for statistical genetics analysis, how could we store the haplotype data? The classical data we are encountered with is this data. We use the 0-1. Oh, sorry for this. 0-1 or 1-1 to represent the possibilities about the haplotype result, and this one should be 1-0. No, 0-1. And and these results are stored to conduct the further association analysis. So the association analysis only use the information about 1, 0, but under these circumstances with the VCA format, how could we incorporate that? So we use the 0 to define the reference allele, which is for example, at the first line here, at the position 10375 of chromosome 10, the reference allele might be represented as a TAACC, which we use the 0 to represent it. And it got three alternative allele. So how could we encode the three alternative allele? We use three numbers to incorporate them. So basically, we use 1 to a positive real number to represent the alternative allele. So for this toy example, we got 0. We got 0 for the reference allele as the TAACC, and we use the 1 for the alternative allele, the first alternative allele, which is the alternative allele 1, the star. So it might happen sometimes that we got a star as the sequencing result because we did maybe the result encountered some sequencing problems or didn't pass some quality control issues, and we also use the 2, or we use number 2 to represent the second alternative allele as the T, and we use number 3 for the third alternative allele as AAACC. So you can see that based on this, how many combinations could we encounter? We might have the haplotype as 0-1 or 0/0 or 0/1 or 0/2. So 0/1 means the reference allele, both of them are reference allele. So this haplotype might be considered as the homozygous reference, and 0-1 might be considered as the heterozygous variance, and also the 0 oh sorry. Yes, and 0-2 might be another one, which is actually the reference allele is TAACC, while the alternative one is T. And for 0-0, it's both TAACC and TAACC. And 0-1 might be as the TAACC and star. And similar things might happen for 0/3. And then it also happens that we could encounter 1/1, which is the star/star, and also the 1/2, 1/3, and 2/2, another alternative heterozygous, another homozygous alternative, and also 2/3, and also 3/3. So these are all the possible results. So But now this is the problem. We cannot do the association analysis, even though the most simple one as the trend test, just based on the data more than 0 and 1. So we have to do some data transformation based on the VSIF file into only into data format only containing 0 and 1. Actually, now researchers have created a lot of tools to do so. These tools are generally called as the VCF tools. If you can type it on the Google. Yes, we got a lot of VSIF tools here, but the problem is that even though we start from the same original VSIF file, if you use different VSIF tools, you might get different results for the transformation of the data format. So it's really troublesome. Actually, our lab is currently developing a new data format called as the GDS file format, and I think we would touch that later. But for now, we have to get familiar with the VCF file. So this is the problem. This is first the problem of the VCF file. It's hard to be incorporated into the association analysis directly. And if you remember the first 8 column, after the first 5 column, the following part could be divided into 2 main parts. So the green part is the individual information, which is the individual's sequencing results. So for example, this is the first sample, the second sample, and the third sample. And the format tells you what kind of information was stored in the sample columns. For example, the GT refers to the genotype, and the AD refers to the allele depth. So I think I got a slide. Yes. Allele depth, and the DP refers to the individual steps of the allele. The GQ is the genotype quality for the sample data, and the PL is also another genotype quality for the data. So you can see that the individual data is stored exactly as the data structure stated in the format column. Yes, the GQ and the PL are the details about the individual's genotype quality. And if the GQ is larger than 20, we have more than 99% sure that the sequencing quality for this individual data is reliable. All right, this is for the individual part. And before the individual parts, we got the combined information about this specific position across all the samples. So from the filter part, it is actually the original quality control procedure, original quality control result of the sequencing caller. So if it's annotated with the pass, that means this position has already passed the original data quality control algorithm, and something they have conducted something similar to the Harding-Weinberg test, and if it failed to pass that kind of test, they would have different annotations here. And this QUAL is actually the quality score for the specific position. And in this info column, you can get all the necessary info for this specific position about this SNPs or the mutation. So you can see that the AC reference to the allele counting genotype, QD reference to the variant quality by depth, the DP is the combined depth, and the AN is the allele number, and the AF is the allele frequency, which you could use to calculate the minor allele frequency. And you could incorporate all this data into conducting the Harding-Weinberg test. And you could also use the individual data to help you to detect whether this mutation is MMP, insertion, deletion, complex, symbolic, star, or unknown. And you could use the information to help you to decide whether this SNP is transition or transversion, or they are heterozygous, homozygous reference, or homozygous alternative. And one more thing to address is that you might encounter something as the dot/dot. So that means this data is missing. All right, that's the basic overview of the VCF file. Well, it's really interesting to go through. It's hard to go through all the details, but even though it's very interesting, I think. So feel free to talk with me about that after the course. And Yes. So the first disadvantage we have addressed about the VCF file is that it is hard to be transformed into a unique format for conducting the downstream association analysis. And another trouble might be the space consuming issue. So if you could change the VCF file into other different data format, it could save you a lot of space. This is the new data format our lab is actively developing now. And if you use the GDS instead of the VCF file, you could make the compression rate as 0.0001. So it is really an efficient way. Yeah. If you are interested, feel free to get in touch with me about that. All right, then it's the third part. So the third part is basically about the ChIP-seq motif analysis. I would like to go through the analysis procedure in this homework, and when we go through the homework one by one, I think you could control all the techniques for analyzing the ChIP-seq motifs. So I have talked about the question one in last lab, so I want to jump this part. So basically, it's just doing some BWA mapping based on a toy example we have created on the Odyssey. And I also I also prepared the SAM tools here for your references to help you to calculate the what are we calculating? To calculate the successfully mapped proportion and also the uniquely mapped proportion here. And for the second question, it's asking you to do the quality control by using the max2 filtered up command based on the toy example we prepared. So this is conducted based on the results you obtained from question one, which is the SAM file here. And the SAM file from question one should be the input for the question two here. And you might got the bed file as the quality control procedure. You can check as I mentioned in last lab, you can check the error file for the redundant percentage. And for question three, now we are going to do the ChIP-seq part. So it's not the toy example anymore, but the entire data and the tumor.bam and also the normal.bam. So the first thing you have to do is to you have to do the quality control for both the tumor sample and the normal sample before you do the copies. So to give you more hints about what I'm talking about, maybe you could open our R session. Wait, what happened? Sorry for troubling. Okay, we got it. All right, if you can go to question two. So here, before you do the copy step, you have to do the similar quality control step in two. For example, if you want to do it for this specific file bed, you have to name the file bed here, file bed, and also as your output bed, and then use this output bed as the input for this command. You have to do the similar issue for both the normal sample in question 3 and also the tumor sample in question 3. Yeah. And then for question 4, now it's not comparing according to the background, but just comparing the normal sample and the tumor sample. But remember to do the quality control before you do this sort of thing. So basically what I'm talking about is that before this copy, you have to do the filtered up for the tumor.bed and store it as the tumor.bed and do the similar thing for the let's see, normal.bed and also the normal.bed, and then use the treated one is the tumor.bed and the bad one is the normal double, which is the output here and the output here, and combine them to conduct the max 2 peak analysis. All right, sweet. I think you are you are ready to do the first 4 questions. So question 5 is very simple. If you go to the system database, I have done that thing in last time, but I can do it again. And choose the species and the homospan. And what are we searching for? Oh, the prostate. Okay, let's go to the prostate. Prostate. Oh, here. And what factor are we looking for? It's AR, right? All right, now you get all the ChIP-seq data about the AR for the prostate tissue. So you got around 17 ChIP-seq data results. So for this question, the answer might be, are they over 10? Yes, they are over 10. And for question 6, it's asking you to check the system's data quality. So maybe we can open a new table. Let's do that thing again. This is prostate. Okay. All right. So you can check all the quality control in all the quality control parameters here. Basically, they got 6 parameters for the quality control. First one is the sequence quality. Second one is the mapping quality, and the library complexity, the chip enrichment, the signal to noise ratio, and also the regulatory region. So what you have to do is to see what is this parameter doing. So you have to state that what could this sequence quality tell us about the quality of this ChIP-seq data, and what does this mapping quality tell us about the data quality, the ChIP-seq quality of this data. So uh, as you can observe that the more green you got here, the ChIP-seq data would be more reliable, and the more red points you got here, the ChIP-seq data would be worse. So if it's asking you to pick some good quality ChIP-seq data, good quality. So if it's asking you to pick some good quality ChIP-seq data, you might prefer some ChIP-seq data with all green. And if it's asking you to pick some bad quality ChIP-seq data, you might prefer more red one, such as this one. And if it for I think for next question, it's asking to check the company and the catalog number. So you have to compare some good quality ChIP-seq data and bad quality ChIP-seq data. For example, if you could click this ChIP-seq data, and you got all the details here, and if you can check the GL of that, you can get the information about the catalog number and also the company on this page. So you might prefer something that had prefer the company that generated the ChIP-seq data with all green. Yeah, sweet. Just to play around with it. So that's for the second part. And for question 8, so it's asking you to do, well, now we are going to do analysis about the collaborating transcription factors. We provided a lot of softwares here. Well, the system requires you to do to register for a free account, so I give it up. And the Homer requires you to download the original packages and install it on your own PC, so I give it up again. And what I found really easy to do is the reader. So if you can click the reader and go to the home page, so you just need to submit the bed file. So the bed file here is actually the result from your question 4. Remember your question 4? That's the result for this question. So for question 8, now I have already downloaded my bed file from the Odyssey. Not this data, it's in the work file. So this is my result for question 4, and it's tumor over normal, and I submit this bed file onto the widder. Yes. And then we check the default setting, it's human species, right. And it's HD38, correct. And the background, we don't want other background, and it's just for 2019, right. So we don't have to change the default setting, and we could run it. All right, we have to wait a while. So recall that this step would give us the result for the potential collaborating transcription factors of AR, and this is the first strategy to get the potential transcription factors of AR. Oh, one thing to mention about the next question. So I know that the second question, the question 9 is for undergraduate, while question 8 is for graduate student. But actually, you have to do the first question, then you can do question 9. So maybe undergraduate students also have to do question 8 before you can, if you want to complete question 9, because it requires you to compare the result of question 9 to the result of question 8. It's still running. Okay, now we got the results. So you can see that they got a lot of potential collaborators with AR. How could we? So you have to choose some top candidates for as the potential collaborator of AR. So how could we choose that? We should do that sort of thing based on these three thresholds, three parameters. The first thing is the correlation. So this is the Spearman correlation with the AR. So basically what we want is the highest absolute value of the Spearman correlation. But you can see that there is a p-value followed by this. So what does this p-value mean? So this is the preferred position p-value. It tells you how accurate and how confident you are about the inference at this specific position of this kind of transcription factor. So in other words, it tells you whether you are sure, how confident you are about this specific position to be identified as this sort of transcription factors. So the strategy to choose the top candidates might be that choose the transcription factors with the highest absolute value of the correlation, and also the highest confidence that at this position it could be identified as this type of transcription factors, which means that you should pick the p-values as small as possible. All right, and then you could just list the name of the transcription factors, which is the first column here, as the answer for the question 8. And then for question 9, it's asking you to do the, in the system database. So it's again, come back to the system database and to find the results in these ARs. For example, if you could click a good quality one, and they got the QC and get the top putative targets, you could get the genes. And if you go to the QC motifs, you could get the potential collaborating transcription factors here. Okay, do you see similar motifs? Okay, so that's the comparison between question 9 and question 8. And then I think there's some parts that's asking you to check whether they are exon, intron, or intrinsic regions. So how could you do so? If you can go back to the system database, there's a UCSC browser, and if you click the UCSC browser, yes, this is the result for the AR, and if you move the mouse at this part, this tells you that this part is intron, and if you go one step previous, it tells you that this is exon. So just go through this picture through your eyes to see whether the majority part of this line is intron or exon, and that would be your answer for this question. All right, then it's for question 10. So question 10 is another procedure to find the potential collaborating factors. So this is the timer, which is the sub-project of the system program. If you could go to the timer website, it's also developed by Dr. Liu's lab, and we could hold another lab to talk about the details about how they design the timer with the tumor immune estimation resources. So all the software is developed based on the TCGA data. So so basically they collected all the analyzing results for the transcription factors based on the TCGA data to see whether they have been expressed differentially, comparing the tumor sample and the normal one. And how could we see whether a specific transcription factor could regulate the gene expression within the normal sample and via comparing the normal sample and the tumor sample? We could check it directly from the differentially expressed one. So you could consider the timer as a summary library for the information about the potential transcriptional factors. So at the differentially expression channel, if you type in the target transcription factors, such as for this lab, we are going to talk about AR, and just submit it. Wait, you would got the result here. So you can see that these characters are the represent some specific cancers. And what we are focusing for this week is the PRAD, which is the prostate cancer. So you can see that for the AR, the red color here represents the tumor sample, and the blue color here represents the normal sample. So you can see that basically in for the PRAD tissues, considering the AR situation, the expression of the normal sample is slightly higher than the mean expression for the tumor sample, and though the p-value is not quite significant. All right, then you could, for that question, then another problem might be that how could we compare the correlation between different transcription factors? Because after all, we want to identify the collaborating transcription factors of AR, right? So we have to see the correlation of other transcription factors with AR. So you can go to the correlation channel and you can type in the AR here and also the target transcription factors you want to analyze. For example, if you inherited the analyzing results from question 8, if we want to see the FOXP1 and its correlation with AR, we just type FOXP1 here, and it should be adjusted by the tumor purity, and just submit it. Okay, now from this plot, you can see the correlation between the FOX2, FOXP2, FOXP1, and also the AR, and sadly, it's not quite significant here, and so it may not be considered as an ideal candidate for the collaborating transcription factors. And if you could type in other potential collaborators, such as the FOXD1, maybe let's try it. Submit it. Now the result is quite significant, so it would be considered as some ideal collaborating transcription factors candidates. Okay, that's all for question 10. And for question 11, it's the third procedure for identifying the potential candidates for the collaborating transcription factor. It's the it's the toolkit of the system program. So if you can go to the website. Yes, and just this requires your result from the question 4, which is the finding results comparing the tumor sample and the normal sample. So recall that for this data, this is this one, but the first thing you have to do is to check how many peaks you got from this file. And actually, from this file, you got around 10,000 peaks here. I can take you to go through it. Yes, one row refers to a peak, and so we got around 10,000 peaks here, and we can't use this default setting. We have to change it to 10,004. So, and we choose this file here. And we want to analyze the transcription factors, correct. So we don't have to change other parameters. So just submit it. Okay, just wait. Still didn't show up. Alright, what is expected and the result is this kind of figure. So the factors listed on the first column might be the potential candidates for the collaborating transcription factors. I think it's working now. Well, it must be a busy time. So you guys must be competing with me to run the analysis at this moment, I guess. All right, just forget it. We would go back later. And then it's the fifth part. So for part 5 is actually do the downstream analysis followed by the. Okay, now we got the result. So if you show the results in figure, you could get the similar results as mine. Yes. All right. Then for part 5, the first thing is to for part 5, we want to see the functions about all the potential transcription factors. So the first part is about is to identify. We want to find the. Now we'll try to see what target genes this AR binding sites regulate. So the first thing is to the first strategy is to detect these target genes as the putative AR target genes in the prostate cancer by searching through the windows around the transcription start site. So to do such sort of thing, you have to download this transcription start site annotation results, the reference seq annotation results from the UCSC database. It could be downloaded through this link, and if you can search this file. No, sorry. Oh, interesting. What happened? Seriously? No, not this one. Sorry. It's as the first link. Sorry for that. All right. Now you might got it. Okay, so this GZ file, and if you download it, you would got this compressed one, and just extract the txt file from it from to here, and if you can take a glance at it. So for this question, it's asking you to write a Python code to analyzing the results. If you can take a glance at it, for question 12, I think. So let's check the original data structure. Maybe it would be helpful to just open it as an Excel file. All right, so for here, you can see the original data structure for the reference annotation. And you could do some, for the strategies of writing the Python, you have to identify the transcription start site and then compare it to the annotation results of the base file to identify the nearest transcription factors within 3000 kb of that specific transcription start site. Just pay attention that for the positive strand, you have the start position might be considered as the transcription factor start site, and for the negative strand, the end position would be considered as the transcription factor starting site. So when you are writing Python, just pay attention to these differences. And but if you don't have enough time to write the Python code, I could provide you another backup as the BetTools window command. So BetTools window command could do the window detection around 3000 window detection directly for you. And to do so, you have to prepare the original TXT reference file as a bit file. So as I mentioned, the TSS for the different strands are different. So it's different for genes on positive or negative strand. It's start site for genes on the positive strand, and negative for genes on the on the negative strand. It's the end side, end position of the genes in the negative strand. So you have to do some annotation based on the original TXT file. So for example, now you can see the original data structure of the txt file. So remember the basic columns, basic data structure of the bit file. We only need 3 columns. The first thing is the chromosome. Second column is the start position. The third column is the end position. And we might also consider to keep the information of the strand to help us to identify the TSS and also the gene ID, so which is the reference seq ID. So now we want to keep all these five columns, and you can see now the data structure might be changed to. All right, this is exactly the data structure for the bit file, and now we want to get the TSS for both the positive strand and the negative strand. So we subset them. And for the positive strand, we want to use the start position as the loci of the TSS. So we keep both the start position and the end position as the same for the TSS. And for the negative strand, we want to use the end position as the TSS. So we would keep both the sorry. Okay, so we will keep the start position and the end position the same as the end position. And to ensure that the end position is larger than start position, we just use the command as the TSS new end as the start plus 1. So now if you can use the head of the TSS new, you can see that the end position is 1 base pair larger than the start position. And then stored the TSS new database as the TSSG 38-bit file, and then submit it to the Odyssey. So now we could do the bit tools analysis on the Odyssey. So this is the main part. Before we do such kind of things, do remember to apply for an interactive session. So with this command, I have already applied for one. So, but this might be out of time now. We have to do it again. Sadly, new window. All right, and apply for an interactive session. And in order to use the bit tools, we have to load some pre-requirement packages. So remember, how could we check whether we could load bit tools? We could use the module spider bit tools to see whether bit tools is available on Odyssey, and also other pre-required modules to be loaded before we load the bit tools. So I just jumped these steps because I already know how to load them in the last lab. And now the first package to load is the CentOS 6. A little bit slow. Just wait a while. Wait. Okay, great. And then it's the bit tools. All right. So now we can detect it. I can go to my home address where I store all the results. So I submitted the TSS file, HG38 file as the workplace as this one. So if you can check this one, it's exactly the same as my R output here. And then I also stored my result from homework for question 4. Sorry for the mistake here. It's question 4, not question 3. And I also stored it here as this file. So this is the result for the this is result for the uh, transcription factors binding peaks, and then we could do the we could identify the target putative genes. So the first part of this command before this larger sign means that we want to identify the, if you just type in them, you could get all the results here. So all of them are the putative target genes. And if you want to store them, just use the whole command. And it stores them at the pass after the larger sign to this target file. And if you can check the target file, so at this position. Yeah, these are all the putative target genes with the gene ID. And if you want to count the number of the putative genes, now you can get the result here. It's around 1,300 or so. All right, great. Then for question 13, it's asking you to overlap the putative AR target genes you get and also the differentially expressed one. So we have prepared this file on the Odyssey. If you can go to our course folder, HW4. Yeah, this differentially expressed the data is here. And what you have to do here is to calculate, the first thing is the AR target genes from binding unknown, so which is just the putative AR target genes you get one step further, which is the 1300 genes you get from question 12. And then the second part is to do the overlapping of these 13,000 genes, 13,000 putative genes, and also the genes listed in this differentially expressed file. So you have to do some overlap calculation based on these two files. I provided you an R session to do so, and if you want to do such sort of thing, you could download this putative AR target genes. Well, originally it's a bat file, but as I mentioned, bat file is actually a TXT format, so you can change the name as the just change the name as the .txt and then read it in and give them some call names. And oh no, sorry, this is the wrong one. I have to download it. Stat115 HW4 work. All right, so it's here. Remember how you download it? It's stored in data, right? Oh no. Where did I store it? Oh, it's in Lab8. Yes, it's in data. So this one is out of date now. Move to trash. And I have to open a new window. And this file, where you want to store it on your own PC. And great, now you get it on the data folder, and you can just change the name as a txt, use txt, and then we can read it in. Yes, now the number is the correct one, around 1300, and then read in the differentially expressed data, which we get from the folder in our website here, in our Odyssey account here, this one. Then you can merge them to get the overlap. All right. Great. Then for question 14, it's doing the overlap of the top 500. So question 14 is also similar to question 13. It's also doing some overlapping issues, both with the differentially expressed one and without the differentially expressed one. So with the differentially expressed one means that you have to do the overlap between the transcription factors binding result and also the ARP regulatory potential file. So without differentially expression means that you do not need to do the overlap step. And for question 15, let's do some comments. So you don't have any instructions on this question, I think. All right, that's all I have prepared for this lab. And I have some acknowledgements and good luck for your homework. And do remember to submit the homework in time for this time. Good luck and have a nice weekend. See you next lab.